Welcome to the CRISPR-Cas video series from the project Genetic Engineering and the Environment. Today with the first part of the risks. In this video, I will talk about unintended effects on the metabolism of plants and their interactions with ecosystems that can be caused by the genome editing technique CRISPR-Cas. My name is Katharina Kawal. I have a doctoral degree in molecular biology and I will guide you through this video. Plants interact with many other species in their respective ecosystems. This can be plants of their own species or plants of a different species. In addition, plants are part of a food web and interact with various animals. Plants have a specific composition of microorganisms that colonize the plant both above and below ground. This is also known as the plant's microbiome. The climate conditions influence how the plant develops and can trigger stress reactions in the plant. By using genome editing, new traits can be generated in plants which can intervene in these interactions. Although the new trait itself may cause disruption of these interactions, disruption can also be caused as a result of the CRISPR-Cas process. We have to take a closer look at the level within the plant on which the gene scissors work to illustrate the different risks of CRISPR-Cas. On the molecular level, on the one hand, the intended changes at the target site can cause unexpected effects. That means that the use of the gene scissors is successful at the DNA level, but that success comes with unintended side effects. On the other hand, unintended changes in the genome through an incorrect or imprecise application of CRISPR-Cas can occur, causing errors in one or more parts of the DNA. Both aspects can give rise to risks for the genome-edited plant and for its interactions with the environment. In this video, I will explain the first aspect in more detail, and in the next video, I will discuss the second aspect. In the video about the possibilities of CRISPR-Cas, I showed you that complex applications of the gene scissors generate new genotypes by SDN1 applications. The complexity increases the likelihood of unintended and unwanted changes in other metabolic pathways. For example, if a gene that is involved in several processes in the cell is knocked out, some of these processes may be unintentionally disrupted. A more detailed explanation of how new genotypes can arise through complex applications can be found in the video about the possibilities of CRISPR-Cas. The unintended effects of the intended changes at the target site in the genome can be diverse. The development of the genome-edited plant could be influenced. For example, it could grow slower or the leaves could have a different shape than non-genome-edited plants. The composition of the plant could be altered, such as the composition of fatty acids or other metabolic products. The plant's reactions could be altered under stress conditions. If a plant is exposed to heat, for example, it reacts to the environmental conditions in the short and long term by regulating a large number of genes. Unintended alterations in metabolic pathways could, for example, result in fewer signaling molecules being formed in the genome-edited plant, which can influence the regulation of the stress response. In addition, the intended changes could lead to an enhanced fitness, which brings the genome-edited plant a survival advantage and makes it more invasive. The just mentioned aspects can have potential unintended and unexpected effects on the ecosystem in which the genome-edited plant grows. Here you can see again the genome-edited plant from the beginning in its natural environment. The intended changes in this plant could affect the defense mechanisms against pests or infections with plant viruses or bacteria. The plant could therefore become more susceptible to pests or pathogens. In addition, the communication between individuals of the same species could be altered if, for example, fewer precursor molecules are produced for the formation of certain messenger substances. Plants can use such messenger substances to warn each other against pests, for example. The same applies to the communication between plants of different species. The intended changes could have unintended effects on the relationship between plants and pollinators, as well as interfere in the existing food web. The intended changes could also lead to the formation of toxic or allergenic substances. For example, a genome-edited plant that is a food source to an insect could become toxic to that insect, and that insect is no longer available as a food source for the animal that ate it, causing a disruption of the food web. This would be an unintended consequence for the ecosystem. Additionally, the composition of the microbiome can be altered. And as already mentioned, the genome-edited plant is exposed to changing climate conditions, which could also have unintended effects on the just mentioned reactions of the plant. Whilst all these risks are plausible, there is currently little knowledge on how they might affect the ecological interactions and safety of genome-edited plants. It is therefore necessary to subject genome-edited plants to an approval process and to investigate their risks. In summary, the intended changes at the target sequence induced by the gene scissors can generate plants with new traits, but can also affect multiple biochemical pathways. If, for example, a gene that is involved in several processes in the cell is knocked out. The release of such genome-edited plants is associated with risks for the ecosystem. This underlines the necessity of regulating genome-edited plants to investigate these risks and to prevent unintended and unexpected effects at the ecosystem level. This necessity also applies if only small and few changes in the genome of plants have been induced. In the next video, I will explain further risks of CRISPR-Cas. I will show you which unintended changes in the genome can be caused by the use of CRISPR-Cas and the use of old genetic engineering methods. Thank you for your attention. You can find more information at the website of the project Genetic Engineering and the Environment.